Hello everyone. Today I will discuss an important topic of molecular biology, nucleosome packaging and chromatin organization. So let's start. In eukaryotic cells, approximately 2 meters of DNA must fit inside a nucleus of only about 6 micrometers. This enormous compaction is achieved through hierarchical DNA packaging. The fundamental repeating unit of chromatin is the nucleosome. A nucleosome contains around 147 base pairs of DNA wrapped approximately 1.65 times around histone octamer. The histone octamer consists of 2 copies each of H2A, H2B, H3 and H4 histones. Linker DNA connects adjacent nucleosomes, and histone H1 stabilize this structure to form a chromatosome. Thus, nucleosomes not only compact DNA but also regulate DNA accessibility and gene expression. Levels of DNA packaging. DNA packaging occurs in multiple hierarchical levels. First, the DNA double helix has a diameter of about 2 nanometers. This DNA wraps around histones to form the 11 nm beads-on-string structure. Further folding generates the 30 nm chromatin fiber. The 30 nm fibers organize into loop domens of approximately 300 nm, which attach to scaffold proteins. Further condensation produces the 700 nm chromatid and finally the 1400 nm metaphase chromosome. This progressive packaging ensures efficient organization, protection and segregation of genetic material during cell division. 30 nm fiber models. The 11 nm nucleosomal fiber undergoes further folding to form the 30 nm fiber. 2 major models have been proposed for this organization. The first is the solenoid model. In this model, adjacent nucleosomes interact with each other and linker DNA bends inward. Approximately 6 nucleosomes occur per turn. The second is the zigzag model, where alternate nucleosomes interact. Here, linker DNA passes through the central axis of the fiber. Histone H1 plays a critical role in stabilizing higher-order chromatin structures. High concentrations of H1 favor the zigzag arrangement. Both models explain chromatin compaction and remain highly important for CSIR-NET examinations. Histone tails and their functions. Histone proteins possess flexible N-terminal tails that protrude outward from the nucleosome core. These tails are extremely important because they regulate chromatin organization and gene activity. Histone tails help stabilize interactions between neighboring nucleosomes and promote higher-order chromatin folding. Additionally, they undergo post-translational modifications such as acetylation, methylation, phosphorylation, and ubiquitination. Another important function is that nucleosomes wrap DNA in a left-handed manner, introducing negative supercalling, which facilitates processes like transcription and replication. Histone code hypothesis. The histone code hypothesis proposes that specific combinations of histone modifications create a regulatory code that determines chromatin state and gene expression. For example, acetylation of lysine residues usually activates transcription by opening chromatin. Methylation may either activate or repress transcription depending on the modified residue. Phosphorylation is commonly associated with chromatin condensation during mitosis, while ubiquitination participates in transcriptional regulation. These modifications are recognized by specific effector proteins that recruit transcription machinery or silencing complexes. Therefore, histone modifications function as epigenetic signals controlling genome activity. Euchromatin versus heterochromatin. Chromatin exists in 2 major forms, euchromatin and heterochromatin. Euchromatin is loosely packed, transcriptionally active and usually replicates early during S phase. It is enriched with activating histone marks such as histone acetylation. In contrast, heterochromatin is highly condensed, transcriptionally inactive and replicates late during S phase. It contains repressive marks such as H3K9 methylation and DNA methylation. Heterochromatin can be constitutive, which remains permanently condensed, or facultative, which can reversibly switch between active and inactive states. Thus, chromatin organization directly influences gene expression. Nucleosome remodeling complexes. Chromatin structure is dynamic and constantly remodeled by ATP-dependent chromatin remodeling complexes. Major remodeling complexes include SWI-SNF, ISWI, CHD and INO-AT families. These complexes utilize ATP hydrolysis to slide nucleosomes along DNA, eject histones, exchange histone variants or reposition nucleosomes. This remodeling changes DNA accessibility for transcription factors, polymerase and repair proteins. Therefore, chromatin remodelers are essential for transcription, replication, recombination and DNA repair. Histone modifications and gene expression. Gene expression is strongly regulated by histone modifications. Enzymes called writers add modifications, whereas erasers remove them. Histone acetyltransferases, or HATs, add acetyl groups to lysine residues. This neutralizes positive charges on histones, weakens DNA-histone interactions and opens chromatin. Histone destylases, or HDACs, remove acetyl groups, leading to chromatin condensation and transcriptional repression. Similarly, histone methyltransferases and demethylases regulate methylation states. Thus, chromatin structure acts as a molecular switch controlling transcription. Experimental techniques. Several experimental techniques are used to study chromatin organization and nucleosome positioning. Micrococcal nuclease digestion selectively cuts linker DNA and leaves nucleosome-protected DNA fragments of approximately 147 base pairs. Chromatin immunoprecipitation, or CHIP assay, detects interactions between proteins and DNA in vivo. ATAC-seq identifies open chromatin regions across the genome. DNA footprinting reveals DNA regions protected by bound proteins. These techniques provide important insights into chromatin accessibility and epigenetic regulation. Epigenetic regulation. Epigenetics refers to heritable changes in gene expression that occur without altering DNA sequence. One major mechanism is DNA methylation, especially at CPG islands, which usually represses transcription. Genomic imprinting causes parent-specific gene expression patterns. Another example is X chromosome inactivation in females, where one X chromosome becomes condensed into a bar body. Histone variants also contribute to epigenetic regulation by altering nucleosome stability and chromatin properties. Thus, epigenetic mechanisms regulate development, differentiation and disease processes. Nucleosome positioning at gene promoters. Nucleosome positioning plays a crucial role in transcription regulation. Gene promoters often contain nucleosome-free regions near the transcription start site, allowing transcription factors and RNA polymerase to bind DNA. The +1 nucleosome is usually positioned immediately downstream of the transcription start site. Chromatin remodelers and histone modifications continuously regulate nucleosome positioning. Accessible promoters facilitate transcription initiation, whereas blocked promoters repress gene expression. Chromatin states in transcription. Chromatin structure dynamically changes during transcription activation. Initially, chromatin exists in a condensed and inaccessible state. ATP-dependent remodeling complexes reposition nucleosomes, and histone acetylation opens chromatin. This relaxed structure allows transcription factors and RNA polymerase to access DNA and initiate transcription. Conversely, Histone decetylation and repressive methylation marks promote chromatin condensation and transcriptional silencing. Therefore, chromatin structure acts as a central regulator of gene expression. Centromere and CENPA. Centromeric chromatin contains a specialized histone H3 variant known as CENPA. CENPA replaces histone H3 within centromeric nucleosomes and is essential for kinetochore assembly. The kinetochore is a protein complex that mediates attachment of chromosomes to spindle fibers during mitosis. CENPA contains an extended N-terminal region that recruits kinetochore proteins such as CENPC. Loss of CENPA disrupts chromosome segregation and leads to mitotic defects. Summary of key concepts. To summarize, the nucleosome is the fundamental unit of chromatin organization. DNA packaging occurs through multiple hierarchical levels leading to metaphase chromosome formation. Histone tails and post-translational modifications regulate chromatin accessibility and gene expression. Chromatin remodeling complexes dynamically reposition nucleosomes using ATP. Epigenetic mechanisms such as DNA methylation and histone modifications control cellular identity and transcriptional programs. Finally, specialized histone variants like CENPA are essential for chromosome segregation. Before ending, let us revise some important CSIR-NET concepts. A nucleosome core particle protects approximately 147 base pairs of DNA. DNA wraps around the histone octamer in 1.65 left-handed turns. Histone H1 acts as the linker histone. The 30 nm fiber is explained by solenoid and zigzag models. CENPA is variant of histone H3 present at centromeres. Histone acetylation generally activates transcription, whereas heterochromatin formation represses it. SMAR regions are at rich DNA sequences associated with scaffold proteins. These are frequently asked concepts in competitive examinations. Thank you.